Good morning. I was sort of struck by the number of people at the meeting and imagining each of us were a major capsid protein assembling into a particle of a sort, and how wonderful that is to be in a community where people can celebrate science and technology. I'm particularly struck by this, having as a graduate student in 1997, the benefit of participating in the 15th phage virus assembly meeting organized by Professor Callender and colleagues. So to be able to come back 15 years later and hopefully give back a little bit of my own excitement for what we're doing and what I'm learning from each of you is really an awesome opportunity. I decided to cut one of the topics, so I'm not going to talk about our work with PHYX or M13. I'm going to talk about some of our work with serine recombinases and also with translational coupling. I'm an engineer. That's what this slide is meant to represent. What that means is if I say something and you can't understand why I'm interested in something, why I'm super excited about it, just remember this. I like to build things. So I love people who do science and want to understand stuff. That's not me. I'm not against that, but I like to make things. The other reason I wanted to share this is a photograph from a few weeks ago at Stanford, where I am. And Stanford has just started in the last few years a new department called bioengineering. Bioengineering's been around for a long time, but it's new to the Stanford campus, and it's the first new department at Stanford since computer science was started about 40 years ago. And the building here will be the home. The steel finishes tomorrow for the new bioengineering department. So here's the context in bioengineering and at least how I think about it, and why Stanford is investing in trying to make the engineering of biology easier. This gentleman hiking is trying to bridge a gap. The gap here is about 4 meters in length. I think about a different type of gap in genetic engineering. Most of biotechnology that's based on recombinant DNA and synthetic DNA that's delivered to market today is typically encoded by tens of thousands of base pairs of designer DNA that might have dozens of functional components. On the other side of this gap, we have ongoing geometric improvement in DNA synthesis and assembly capacity. So Ataya, for example, at Mitsubishi has been able to stitch together 8 million base pairs of DNA, recapitulating 2 natural genomes into a composite genome that's capable of replication. 8 megabases of DNA construction. That's thousands of functional genetic components. I don't know how to design 8 million base pairs of DNA. The people who are resynthesizing genomes aren't designing genomes yet. They're just sort of copying what nature gives us. But if you think about the numbers, 8 megabases divided by 20 kilobases is a factor of 400. I'll call that the biointegration gap. If any of you could teach me how to get 400 times better at reworking the molecules of life to make new useful biological systems, I could become 400 times better as a would-be biotechnologist. Right? I don't really know what the right side of this gap means if we realize it. And to share with you a way of thinking about it from literature, I just want to give you examples of artifacts, what you can encode with tens of thousands of characters, like a presidential address, or an editorial in a newspaper, or a research report in science. Tens of thousands of characters, including white spaces. But if I were to give you millions of characters, you could do things like a one-act play from France, right? Or an American novel, right? Or a Russian novel. Can all be encoded with millions of characters. So if you imagine what bioengineering looks like, if we could bridge this gap, if we could put together thousands of functional genetic elements, we're probably going to live in a world, if we can pull it off responsibly, that the types of artifacts people make are very different from short declarative statements, make lots of a biofuel or make some recombinant therapeutic protein. For example, as a civil engineer growing up, I was particularly struck by this image from India, where a suspension bridge, a double-decker suspension bridge in this case, had been grown in place from the roots of a rubber plant. Fixes carbon as it grows, it gets stronger over time. Very different manufacturing capacity. This process is guided by humans shaping this, but there's no reason to imagine that you couldn't eventually have autonomous program pattern formation to make things like this become true. 15 years might sound like a long time to make a bridge, but in San Francisco, that's very fast for making a bridge. So maybe we live in a future where you have program pattern formation in gigantic gourds that differentiate into 4-bedroom, 2-bath houses. I don't know, right? But if we get much better engineering biology, we probably surprise ourselves. How could we do it? Don't know. Someone in this room probably knows an older version of this quote, but I was struck by a colleague at Stanford, Marcus Covert, where he observed phage are nature's genetic engineers. They get into cells, they reprogram the cells, and things come out. And they're doing it on a scale of up to hundreds of components. It's pretty impressive. So maybe one way to think about this community connecting with the bioengineering community is you can teach us a lot about how to be more holistic in partnering with biology to make things. So here's the first story. This story came out in PNAS US about 6 weeks ago. And I want to go through it in some detail just to give you a sense of how we think when we get into the weeds of trying to make stuff. Excuse me. Tone is from Thailand. He's a PhD student. And Jerome is from Montpellier, France. And he's a postdoc. Neither of them are bioengineers to start with, but they are now. And if anybody in France would like to hire Jerome, he really wants to come back to France. So please hire him. They want biobits. They want to store modest amounts of information inside cells. I don't really care why they want this. I kind of want it too. If I had to make up stories, I'd say we're going to track cell division events, and the cells will tell you how old they are. Or we're going to automate lineage tracking, so you don't have to look at the stuff under a microscope. The record keeping is in the DNA, and you just sequence it out at the end. Or maybe you want to reprogram lineage and developmental biology in some way. There's a lot of reasons why you'd want to have a little bit of state data storage inside living systems. So this would be a byte. This would be 8 bits, binary digits. And what Jerome decided to do was to establish a data register in DNA. And the reason he wanted to do this is what an engineer would call non-volatile data storage. Non-volatile means the register will hold state in the absence of energy, right? Like a USB flash drive doesn't lose the data when you depower it. In this case, it's going to be data storage in the absence of heterologous gene expression, that once you set this register in the DNA, you don't have to express any proteins and it'll keep the bit set as it is. So we call this thing in the lab, we call it the flippy. Didn't publish it with that name, but that's what we call it, the flippy. And what we're going to do is we're going to flip it back and forth and take advantage of the fact that DNA has a digital, if you will, binary digital orientation, 5 to 3, and you can flip that. We're using long ago established biochemistry that says if you take these BP sites and you have them being opposing each other in one strand, you'll get this inversion. And the way you can imagine this working is if you have the flippy pointing left, it'll be a 1, and that could make a red protein. If you have it pointing right, that'd be a 0, make a green protein. How do you get this thing to flip? You take these integrases. We love serine integrases, as you'll hear more about and already have, because they seem to not require cofactors. That's why we chose them. We wanted to establish data registers that, as I'll show you here, work in E. coli, but gosh, we'd like to get them working in plants and people and worms and whatever. Right? We want portability across kingdoms. So we're going to take an integrase, and we're going to give a little pulse of expression of that and hope this register flips, and then when that pulse goes away, we want the register to stay put. We really want unidirectionality. So here's what cells look like in the green state, microscope on the bottom, 2-color cytometer in the middle, pulse of integrase, quite a long pulse, I'll come back to that later. All the cells switch, goes up to the red zone in the cytometer. And importantly, when we remove the input pulse, the cells stay in the red state. They've switched and now they're holding. Right, so that's memory, when they hold when the pulse goes away. Great. Now we need to flip back. Take the same protein, the integrase, and use this RDF, the recombination directionality factor. Did I get that right? Right, RDF, yeah. Sometimes called an excisionase, but I've learned it's an RDF. And you get both of these proteins and it reverses the reaction. Hopefully. We really want this to be unidirectional backwards, right? So that we get writing and erasing, writing and erasing. Gosh, we were super stoked when we saw this paper from Professor Hatfield's lab, because it details how GP47 from the Bronx bomber phage, BXB1, can be used to reverse this reaction. And figure 5 in particular, the left here was just our favorite figure. This leftmost band shows the absence of excision if you don't have the RDF in vitro. And then, as you add lanes 1 through 5, increasing amounts of this RDF, you get more and more excision, I think, if I remember correctly. And we didn't spend much time looking at this figure. We just loved the fact that as you added more RDF, these bands went from not excising to fully excised pretty quickly. And it looked like it was unidirectional. Much later, when you'll see we had a lot of trouble moving this inside a cell and getting it to operate as we liked, I went back and looked at this a little more carefully and replotted what the equivalent concentrations of the proteins would be inside a cell. And it turns out that leftmost lane, well, that's 0 RDF per cell, but then lane 1 is about 900 molecules per cell, I'd estimate. That's quite a lot. That's almost at the level of RNA polymerase, if the cell's doubling every hour. And lane 5 is about 5,500 proteins. That's a lot of proteins in E. coli for one protein. Why did that matter? Well, we got to get cells to go from the red state to the green state. And when we first tried to co-express the integrase and the excisionase, or the RDF together, we got yellow, which meant that they were going back to green and then back to red, it looked like. And we could split those populations and isolate it and so on, right, where you'd see weird things happening in the absence of the pulse after we tried to reset it. The solution, well, it's from the in vitro biochemistry. We just have to get enough excisionase made to drive the reaction backwards. So we're eventually able to do that by overexpressing, if you will, the RDF. And that cleaned things up. And we could mostly, never quite perfect with this reset reaction, we could mostly get a full reset. You see one cell here on the micrograph is red. Okay, so we're pretty happy with this. What I've just showed you, the set and the reset operation using these serine recombinase functions, those were established in independent lineages, not in the same cell. So to make a full bit in one cell, we got to get set and reset working together. This turned out to be a nightmare. All right, here's why. So imagine you're doing a reset. Not only do you have to get the stoichiometry of the proteins right, you have to get their dynamics correct. For example, let's say you've just done a successful reset operation, but now the excisionase gets degraded first for some reason. You're now left with the integrase by itself in the cell. That corrupts the reset. The reset now reverts to being a set operation, and you get bidirectionality. How do you solve this problem? You stabilize the excisionase and you overexpress it even more. And that solves the problem. Except that expression cassette for the excisionase is now so powerful and the protein is so rock solid that little sputtering, noisy transcription that's always sort of going around the chromosome will lead to spontaneous accumulation of the excisionase, such that the cell always has the RDF, which means that when you go to try and operate your set reaction, you already have the excisionase. So your set reaction is no longer going to work. It's going to spontaneous reset after you set. How do you fix that? You underexpress the RDF and you destabilize it. Well, wait a minute, that's what we had to not do in order to get the reset working. You see where this is going? The two functions, set and reset, might not be compatible within the same reaction volume, the same cellular milieu. All right. Jerome thought he was coming to a really hot genetic engineering lab when he moved from France, and this would be all very easy. You know, we're just trying to express a couple proteins and get them to behave. These are complicated plots, so I'm going to skip them. You can read about them in the paper. What we really needed to end up doing was to control the synthesis and degradation of a couple proteins to within a factor of 2 rate. That's how much precision we ended up needing around the rates of synthesis and degradation of these proteins. Precise engineering is the central dogma. But from an engineer's perspective, we don't really have any tools to do that. When we go to engineer heterologous gene expression, we're typically using found objects harvested from nature that have never been regularized to support precision reuse. Right? It's like building a wall from rocks that you find in nature. Right? You can do it, but it's craft. Thus, it took us 750 attempts, and by attempts I mean different DNA designs tested in the lab to see if it would maybe work, in order to eventually get a working system. And this is what the working system looks like. So if cells are in the set state, we can reset them, they'll hold. This is looking on the y-axis here, and just one of the fluorophores green, and so on, and we can repeat this. These inputs, by the way, are for 10 cell division lengths, right? So quite a long input pulse. If we want to have a cell division cycle counter, we have to be switching this thing much more quickly. So we tried that, and this is as fast as we've gotten this thing to work yet. This is with input pulses for the set and reset that are 2.5 cell division events. And the target you have to hit in translation and degradation rate control gets smaller and smaller and smaller as you try and run the system faster. You need more precision to get faster switching. And I'll just point out that the reset with the RDF is not as clean, again, as what we could see with the integrase alone. A couple observations about this system. This is what it looks like at the DNA level, the architecture. There's no feedback in this system. So there's no formal bistability. There's no fancy control architecture. The fact that this thing holds state at all is pretty remarkable. It's a very reliable data register in the absence of anything happening, right? And to quantify that, that means there's not the spontaneous switching rate is less than 1 in 10 to the 12th cell division events, with no weird feedbacks or anything like that. So I think we could make this faster and better by layering on some of the architecture you see in natural systems. Another thing is, if you look at the details of how this ended up getting to work, you'll see some odd things like GTG start codons. Odd for a genetic engineer sometimes, but not odd for a phage geneticist, right? We're using non-consensus SSRA tags to control differential proteolysis of some of these proteins on the reset, and so on. Okay, so now we want to go to 8 bits, or 1 byte. I want to make things like this. This would be a synchronized ripple counter, where you have the lowest bit on the left and then just driving each bit over and over and over again. To do this, we don't need a latch. We're going to now need toggle switches, which need 2 latches per toggle. We need 8 toggles. So this is why at the end of the paper, as Professor Hatfield showed, I need 16 recombinases. Right? And then I'm going to need to basically synchronize them with additional AND gates, Boolean AND gates. So that means I need a couple transcription factors or something to do that. So this is going to have about 100 moving parts, about the size of a pretty decent phage. And I need to figure out how to get this working. Right, so you can see why this becomes fun, right, and also somewhat challenging. One of the tools, which may seem a little weird to you, and is very controversial in the engineering community, is called abstraction. And this is something that we are trying to steal or borrow or just adapt from computer science and apply it to help manage the process of engineering genetic systems. So this is what it looks like in bioengineering. So let's say on the preceding slide I want an 8-bit synchronous counter. Wouldn't it be nice if I could be working up at that level of function, pretty high level, and think about different types of counter architectures, synchronous, asynchronous, how to connect the toggles, da 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 da. And then when I needed things like a latch, I could just say, hey, Jerome, give me 8 latches. And Jerome would pass up the latches, I put them together, they'd work, right? And let's say Jerome needed a whole bunch of serine recombinases to make a whole family of latches. He'd go, hi, somebody give me 16 recombinases. And here are the recombinases. They don't use the same DNA sites at all. They're all independent, right? And then somebody go, oh, I need the DNA encoding all these recombinases. And somebody just snap, here's the DNA, right? So that's what this is trying to represent, that you could organize the complexity of biology across levels of function such that nobody would have to be an expert on everything, right? Imagine being able to be a genetically encoded data storage engineer making counters and stuff like that, and you wouldn't even need to know that DNA is made up of 4 bases. Or you just compile down to that. Much like, I kind of know that computers are programmed in 0s and 1s, but if you wanted me to write you an email using machine language, not going to happen, right? Because we're compiling down. And somebody knows that, but I don't have to know that. You don't have to know that. The reason this is very controversial, as you can imagine, is it's not true, right? What I just described was not a single call and response between the device level, this latch, and the parts, but 750 call and responses. So that you could make this up as a cartoon, but the interfaces aren't quite worked out. That's why this is tricky. So I'm going to switch into a second story. And this is a story where we're trying to work on the interface between functional devices like latches and parts that implement those systems, and seeing if we can collapse the complexity of that relationship, cut down the number of exchanges between levels. This is a project that I've been very privileged to work on with a colleague at UC Berkeley, Adam Arkin, in the bioengineering department there. And we started a public benefit project called BioFab. BioFab stands for BioFab International Open Facility Advancing Biotechnology, BioFab. It's a recursive acronym, which is something we like very much. This is the team that worked together. These people were very risk-friendly. They took a big chance, basically pulling themselves out of academic environments or commercial environments and trying to work as a professional team, struggling with making parts easier to use. And Vivek Mutalik is the team leader in particular, so I want to acknowledge him and everybody here. So what they started with was trying to debug why engineering things and getting precise behavior has been difficult. One of the things you can observe was when people do heterologous expression engineering, the parts you're using, say a promoter or 5' UTR, are typically not regular in any way. So what that means is if you reuse parts over and over again, you get quite different performance. For example, this is taking 7 favorite promoters for a prokaryote with about 10 or 11 5' UTRs for initiating translation and making all combinations of those to express either GFP or RFP. And the heat map shows across a range of expression how the same combinations of parts produce different levels of expression. For example, over on the right, the second column from the right, you see U3 and P1, that blue square right over there. For RFP, it's quite low expression, but for GFP, it's quite high expression. I care about precision with 2-fold range. This is variation about 30-fold. Why is this hard? Not a mystery, but basically what it comes down to is when we code intentions into biomolecules, the biomolecules have no knowledge of our intentions. They're just molecules. For example, if you think about the initiation of translation, let's say I have a 5' UTR with the Shine-Delgarno core driving expression of a coding sequence, And if I were to claim that I had a standard, really nice 5' UTR for initiating translation, could I actually guarantee that it would behave with any coding sequence for your favorite protein? Or would the translation activity of that 5' UTR change as a function of changes in the local genetic context? Well, it turns out this is obviously true. For example, if we take two different genes of interest and translation is initiated by the same 5' UTR with a sequence of UAAGG, AGGU, the two genes of interest, the top one, gene of interest 2, has a sequence right after the start codon that is a perfect reverse complement to the core of the Shine-Delgarno, whereas the one below disrupts the majority of those base pairs. Thus, when you fold it up in a simulator, you would predict a pretty strong stem loop for the top gene and not for the next. When you actually make this and try and express it, you get no synthesis of the protein from one of the genes and quite a nice bit of protein for the other. So you're trying to reuse a part, but its behavior is qualitatively different. Maybe we could use computers to keep track of all of this and model what's going on. So there's been a lot of work in my community to try and make this true. For example, here's a nice paper where they're detailing automatic design of synthetic ribosome binding sites to control protein synthesis. And just to highlight the main claim in the paper, experimental validation of 100 predictions show that we can accurately design UTRs to initiate translation across a 100,000-fold dynamic range to within a factor of 2. This sounds great. So we tried this out in making that set-reset latch I was introducing before. And when we designed one of the set UTRs for flipping with integrase, we found that it always flipped. We couldn't hold state. It was always translating. So we made a second attempt. That's what the second attempt came out of the computer. It never flipped. It wouldn't make the protein. So we used the calculator to make a third attempt, and the third attempt switched. Great. Well, what's going on? The calculator is not perfect. And the authors are pretty careful about this. If you read into the paper, you'll find, according to the distribution of the calculator's error, an optimized RBS has a 47, call it 50% chance of expressing the protein to within the twofold range of your target. So if you want to get the protein level that you want, you're going to have to try multiple designs and screen. Right? And it cuts the number of attempts you have to do, but it's not perfect. And that means since it's 50-50 likelihood of being what you want, we're going to have to design, build, and test on a single gene basis and a single gene combination basis. Every unique thing is a new experiment. How am I going to get to designing systems with 1,000 genes? Not going to scale. I'm optimistic about these calculators because when you fold the UTR sequences, you can see that the one that's always on is open. The one that's never translating is in a really tight stem loop, and the one that's working has a nice stabilizing stem loop, and then the RBS looks like it's open. So there's got to be a better way to do this calculation. All right, meanwhile, could we do better than this? Could we make a sort of postmodern expression cassette that when you put the parts together, the behavior is always exactly what you want? And how could we learn how to do that? Long time ago, about 10 years ago, we started a project where we redesigned the genome of phage T7. And the reason is summarized in the first sentence of the abstract. Natural systems in biology are selected by evolution to continue to exist and evolve. Evolution likely gives rise to complicated systems, spaghetti code, that are difficult to understand and manipulate. Here we redesigned the genome of T7 in order to specify an engineered surrogate that, if viable, would be easier to study. Let me give you an example of what we're doing. Here's a sequence from the T7 wild-type genome showing the overlap of coding sequences 2.8 and 3, the nesting of that, and the internal gene 3 RBS completely within the coding sequence of 2.8. We had no idea what's up with this. Right? How do we think about this? And we were angry at this type of architecture because it doesn't let us independently manipulate parts. Right? How do I upregulate the synthesis of the endonuclease, gene 3, without at least changing the codon usage of gene 2.8? Right? So really, really annoying to a naive engineer. Thus, we had a designer sequence called T7.1. And this just shows you the sort of thing we were doing. We unstuffed everything. And then we started erasing the legacy elements, for example, in gene 2.8, having to do with the RBS of gene 3 and the start codon of gene 3 and so on. And then we separate stuff out and see how it goes. So this worked when we put it back in E. coli, but the phage grew about factor of 2 slower than wild type. Easier to think about. Maybe we could take that type of approach for engineering standard parts. That doesn't work. I won't show you why that doesn't work, but when you just try and separate everything out and get reliable functional composition, it doesn't work. So I'll call that stupid Endy practice. What we found instead, going back to Yanofsky and Skoner and Makoff from decades ago, is a much more interesting type of architecture for controlling initiation of protein synthesis, which we call a bicystron. And the way this works is if you show up with your gene of interest, instead of putting an RBS right upstream of it to initiate translation, we're going to nest that RBS within a short leader peptide that's about 16 amino acids. And that leader peptide is going to be independently translated by an upstream RBS called RBS1, this orange thing on the left. The idea is that by initiating translation of this leader peptide, we'll regularize any structure specific around RBS2 that might arise due to folding across the junction between the UTR and the coding sequence for your gene of interest. Then the leader peptide and the coding sequence are linked by a -1 frame shift, so a TAA-ATG, and we think that the ribosome is going to stop at that TAA and then reinitiate at RBS2 and off it goes again. But by laminating, if you will, the leader peptide from RBS1 with an active translation process, we're hoping that we won't have any sequence sensitivity to varying genes of interest. So I'll do this pretty quickly. This is a scatter plot showing for 14 different genes and 22 different strength UTRs, the level of protein synthesis we get without the bicistronic architecture, where we're just using the UTR with the RBS of different strengths to drive these 14 different genes. But if we put a upstream RBS there to normalize it, this is the pattern we get. And what this is basically showing, each column is a different gene of interest, and each row is a different UTR. And what you can see is across the rows, the level of expression is normalized. That is, the UTRs are producing the same rate of translation, we think, independent of changes in the protein coding sequence, due to this bicistronic coupling. If you look at it in an absolute sense, here's showing the dynamic range for the monocistronic, meaning a single naked RBS, versus the bicistronic, where we collapse the dynamic range of the proteins, expression rates. This shows in detail now where we use standard promoters, which I don't have time to discuss, and these bicistronic UTRs, promoters across the top, bicistronics along the side. We can regularize expression in detail of two different genes, GFP and RFP, in across about 1,000-fold dynamic range. So I'll just give you a quick comparison. On the left is if you use irregular parts for initiating transcription translation willy-nilly, and that's what the scatter looks like for two different genes, R² of about 0.4. And on the right is with the standardized parts that the biofab team has made with this active translational coupling. This box is the factor of 2 box that I care about and is sort of the benchmark for the calculator. And if I slide this factor of 2 box along the correlation, you can see that most of the points are contained within this box. Not all of them, but about 93% of them. What that means practically, and this shows all the data for all the genes we've tested, is we can now give you standard parts that are reliably reusable for any gene of interest we've tested against, and there's a 93% chance that that protein will be synthesized at a rate within your factor of 2 target window. That's up from a 47% chance. So that's a 750% reduction of error. This is a big deal for us because it allows us to think about designing systems a whopping 10 genes at a time before we have a first expression failure. Now, mind you, we hardly ever know how much of the proteins we want, so we have to get a lot better at designing. But I'm excited about this because as we've listened to the natural genetic architectures a little bit more carefully, we've been able to find ways of getting what we want functionally. Thus, this middle interface between devices and parts is suddenly becoming true for transcription and translation in prokaryotes. I want to quickly introduce you in finishing my talk to the people who are coming after me, because there's a lot of them. These are some of the students we were working with almost 10 years ago, the 16 students, and they were trying to design genetic clocks, and we couldn't get anything to work. In fact, we couldn't even get the DNA made encoding their systems for 7 months. But we had so much fun doing this, and we figured out why it was difficult to outsource DNA synthesis back then, that we taught the course the next year. And we got all the DNA back encoding the student designs within 1 month, and none of their designs worked. But that was so much fun, we didn't even wait to teach the course again. And it went to 5 schools that summer, and one of the projects worked, and they got a nice paper in Nature by taking bacterial photography. Well, that was kind of fun, so more teams joined the next year. And it became known as iGEM, the International Genetically Engineered Machine Competition, sort of genetic engineering Olympics for college undergraduates. It got a little bit bigger, and the students became self-aware, and they made a world map of where they were coming from. And these students, by the way, are doing all the stuff by themselves. It's not core funded, it's distributed funding. And they realize, we're missing some folks, so let's have a special tutorial in China. And so the top 6 schools from China showed up the next year, and they won the competition in 2007. Peking University did. And it got a little bit bigger, and it got a little bit bigger, and it got a little bit bigger. I can't show you 2011 and 2012. Randy Redberg, who's leading this, has done an excellent job of scaling and distributing it. It's now a public benefit foundation called iGEM.org, where teams of students are designing and building genetically encoded systems over their summers, and then they show up and compete. I'll give you a quick example of what they can make. This is from 2009. Cambridge University in England made E. chromai, strains of E. coli that make pigments. Right? These teenagers did this for about $25,000 in capital in 16 weeks. What would you do with this? Well, I don't know. How did they do it? They did it by sharing standard biological parts. They got about half the DNA they needed from other students' projects in previous years. They got free DNA synthesis from a DNA synthesis company, and they got strains over producing carotenoid precursors from DuPont. So this is a do-it-together success. They partnered with the Royal College of Art to imagine what this could be good for. They thought that you could have in the future probiotics that would be instrumented with sensors to detect various disease states of your alimentary canal, such that what comes out of you would indicate which specialist you should go see based on color. Okay, so these people are coming, right? It's not me, it's the next generation of would-be genetic engineers that I think you really want to connect with. They're really incredible and amazing creativity. Last thing. I hope that the future of bioengineering is not merely about the industrialization of nature, and we don't really know how to transcend the default path, but we're trying to lay in preparations. And for example, what we've been doing is paying for residencies between artists and biotechnologists, 1 month in the lab, 1 month in the studio. Sissel Talas from Berlin worked with Christina Agapakis at Harvard, and they were very interested in the microbiology of cheeses, which we've already heard a little bit about yesterday, so I thought I'd put this in. What they noticed was cheeses that are made artisanally have microbes that may or may not be related to the microbes on the human skin. How would you study that? Well, you could go sequence all the cheese microbes and the skin microbes and compare it. Or you could take an engineer's approach, which would be to go around campus and collect microbes from your colleague's skin, go to the farm and get raw milk, and make human microbe cheese, which they did within a few weeks of working together. So cheese made from the microbes in Daisy's armpit, Sissel's nose, Christina's hand, or a philosopher's toe. I happened to be in Boston when they presented this palette of 48 cheeses, and Daisy's armpit cheese is beautiful. It's a citrus floral bouquet. The philosopher toe cheese, you need a new adjective. It's so wrong. Your parents tell you to eat right, you are what you eat. This suggests we eat what we are, that there's a different type of relationship between the natural world and how we might conceive it. So I hope that you have a little bit of insight into the mindset of some bioengineers and what we're trying to do and why. I hope that the future biotechnology does not replace the jungle, but finds new ways to learn from nature and partner in making what we need. Thanks very much.
I don't know what you're on, Drew, but I want some of it. Any questions? A wonderful talk.
When you have a problem with a bad switch, one of the ways of making a better switch is to put 2 switches and to pole them. So what's the downside of doing that?
More parts, right? I mean, in electronics, you do a voting with 3 and, right, or 2, or so, absolutely. But we've got 3 recombinases we're working with now, 3 recombinase systems, and you gave me, if you give me 48, right? Another thing to think about is just latency and burden, right? Every time you're going to express another thing, you put a load on the cell, and so I think it's a good approach, but we were particularly keen to push to see what we could do with the simplest architecture and how far that could go. And what I really have been impressed by is it's a very stable switch without a whole bunch of complexity. And I think there's lessons there for thinking about what natural systems can do.
More questions? I have a question. You are taking a system which is by nature diverse. And the reason we're here is diversity and the mutation rate. And you're asking to restrict that and confine that. We are really going against the flow here, Drew.
I disagree. So I think a lot of technology at a point in its maturation produces a refinement of substrate that appears to be simpler so that you can produce more complicated systems, right? So Legos are pretty simple, but by making the building blocks simple, you get a diversity of possibilities that are easily accessed. DNA is pretty simple, right? Gosh, it's got that nice string of negative charges on the backbone, so it won't collapse up upon itself, so you can swap the bases without screwing up the structure during mutation. But it's allowing all that diversity to happen. So I wouldn't say we're fighting against diversity. We're trying to dance with it in different ways to scale the process of making things. Now, having said that, there are some interesting ideas regarding what you can do to program evolvability. Right, so everybody probably knows that just by remapping codons within the standard genetic code in an open reading frame, you can change by a factor of 2 the likelihood that a point mutation will spontaneously go to a stop. Right, that's your play, just within the standard genetic code. But what if I completely recoded the codons, right, for a new genetic code? How far could I push against evolution? Could I make, for example, could you imagine what I would call a fail-fast genetic code where any point mutation would be selected against? That would be going way, way in the direction you're alluding to. You can imagine it, but you have to go, I think, to a 4-base code. So you have 256 codons. You don't use all the codons. You keep most of them empty. So you keep only 20 amino acids in a 256 code. And that means most point mutations can now map to nulls, which won't be absolutely deleterious, but slightly. Right? So I think there are options in terms of programming evolvability, but I wouldn't represent what we're trying to do is to fight the diversity. Right? We're trying to engage in it.
Just a quick question about the switch. Have you tried to put it in anything other than E. coli?
Not yet. Jerome really wants to do that. And I started talking yesterday, there are people working with spirulina culture in Thailand who want to use it to monitor environmental pollutants coming into open ponds, right? So, I'm hoping people will start to work with that.
Does anybody know of a polymerase that has less of a mutation rate than the standard, what is it, 10 to the 6th or whatever?
Yes, sir.
Is there media dependence on the translation robustness?
Almost certainly. But I don't know if we lose the reusability or rank ordering. It's a good question. I mean, there's so many other context dimensions, right? And Adam's done a lot of work on that. Yeah. I'm sensitive to the fact that we're into the break, so I'd like to propose
we're
I get
fighting
to leave.
nature.
And
So
then I'm happy to talk more.
All right, thank you very much.
I would